Hello A&P students, this is Ms. Richardson. I'm going to be covering the last part of the Chapter 3 lecture. This lecture references the Saladin textbook. I'll also be including some information found in Chapter 4, namely the cell cycle, which is basically the life cycle of a typical cell, as well as the process of mitosis, which is the process of replicating DNA and partitioning that into daughter cells. So it's a form of cellular reproduction. To start off, I'm going to be talking about parts of a typical human cell. Human cells, of course, are eukaryotic cells. That means they have a true nucleus. A true nucleus is a nucleus that's actually surrounded by a double unit membrane. That's in contrast to prokaryotic cells, such as the cells that we see in bacteria and archaea. Those do not have a true nucleus. They still have DNA, they just don't have that double unit membrane that actually partitions the nucleus from the other parts of the cell. So the nucleus in a human cell and other eukaryotic cells is typically referred to as the control center of the cell. And basically, when we say it's the control center of a cell, we're just referring to the fact that cellular activities ultimately derive from the genetic material that's found within the nucleus. The nucleus is the largest organelle typically. It's about 5 micrometers or microns in diameter. And remember that a typical human cell might range from 5 to 10 microns total in diameter. Some cells are actually anuclear. If you see that A in front of a term, think without. So anuclear without a nucleus. An example of that would be a red blood cell. A red blood cell is specialized to transport oxygen throughout the body with the aid of the molecule known as hemoglobin. And it doesn't actually need a nucleus. It doesn't go through the typical mitotic process. It's actually made in red bone marrow. So with red blood cells, they are anuclear. Skeletal muscle cells would be an example of multinuclear due to how they develop in the embryo. We have multiple cells coming together to form a single skeletal muscle cell in a mature human. So we have a multinucleate cell type for skeletal muscle. But typically, most of our cell types will just have a single nucleus. The nucleus does have what's known as a nuclear envelope. That is the double unit membrane that partitions the nucleus from the rest of the cell. And we do have nuclear pores, which are just little openings within that nuclear envelope. That allows for messenger RNA to transmit information from our genetic code to the ribosomes to make proteins. And we also have what's known as the nucleoplasm. There's 2 different components there. The chromatin, which is thread-like matter comprised of the DNA itself, along with associated proteins such as histones. And then we have the nucleoli, which are the dark masses within the nucleoplasm where ribosomes are produced. And ribosomes are synthesizers of proteins. If we were to take a look at a micrograph of the nucleus, a micrograph is just an image that's been taken with the aid of an electron microscope. We see the interior of the nucleus here, the nucleolus in the middle, nucleoplasm, and then the nuclear envelope, that double unit membrane, which will have the nuclear pores in it. So this would be a micrograph of the surface of the nucleus. And when we take a look at cells during our histology labs, we will not see this level of detail, of course. So we have simple compound microscopes, and they provide much less detail, but we'll still be able to see the nuclei in cells. And typically the nuclei will be stained maybe pink or purple or blue, and they will just appear as dark areas within the cell. Okay, so the cytoplasm of a cell is basically everything aside from the nucleus and the plasma membrane. So the cytoplasm will contain most of our organelles. Those are just small parts of a cell that carry out specialized tasks. We have some organelles that are bordered by a membrane, the membranous organelles, such as the mitochondria, lysosomes, peroxisomes, the endoplasmic reticulum, which is often just referred to as the ER, endoplasmic reticulum, and then the Golgi complex. Organelles that are not bordered by a membrane, those are such things as the ribosomes, centrosomes, centrioles, and basal bodies. And then we have the cytoskeleton, which is a system of microtubules and other protein filaments that help to form the shape of the cell. And then we have the cytosol. Cytosol, I mentioned this in class. Another term for this is intracellular fluid. And this is primarily water, of course, but it's not just pure water. It has a number of dissolved solutes in it, such as electrolytes, glucose. So that's going to have its own properties. And then inclusions are things that are within the cell, but they're typically not essential for the cell's survival, and they might just be stored products like lipids, for example. Okay, so the cytoskeleton, basically what this is a collection of filaments and tubules, so it's made out of proteins. It helps to provide the cell with support, organization, and movement. So it's actually oftentimes going to serve as basically a track for various parts of organelles and cell products to move along. It's composed of microfilaments. Microfilaments are typically comprised of actin, which is a type of protein. This forms a network on the cytoplasmic side of the plasma membrane. All that means is on the inside of the cell. The cytoplasmic side would be the intracellular side. So it forms a network on that side of the plasma membrane called the membrane skeleton that supports phospholipids and microvilli and produces cell movement. So phospholipids, again, that's a primary component of the cell membrane itself. Microvilli, not found in every type of cell. They're just small 1 to 2 micron extensions of the plasma membrane. We'll also see intermediate fibers. These help to hold epithelial cells together. Epithelial cells are the cells that will line body cavities and organs. They help to resist stresses that are placed on cells that come from movements within the body, for example. They also line the nuclear envelope, and they help to toughen hair and nails. And then we have the microtubules, which are the largest protein tubules that help to act as kind of a scaffold. So here we have basically a cross-section of cytoskeleton here with all of the various forms of proteins. We have the microtubules. These are these big kind of dark orange tubules that provide more of the overall structure and framework of the cell. We have the microfilaments here, and then the intermediate filaments, which are intermediate in size. So basically, the cytoskeleton helps to provide continuity between the extracellular environment and the intracellular environment, gives the cell its overall structure and shape, and also helps to anchor various parts of the cell, and provides for different parts of the cell to be different cell products, for example, to be transported along rather set pathways. Microtubules are actually, they're not going to be permanent structures. They can actually be kind of spontaneously assembled within the cell depending on cellular needs. Okay, so let's talk about the mitochondrion. Well, mitochondrion would be singular, mitochondria would be multiple, and if there's one organelle that students typically remember from high school biology, it's the mitochondria, because it's known as the powerhouse of the cell. And what that means is it takes the energy in fuels that we consume, like carbohydrates and proteins, and converts it into ATP energy, which is what our body uses to do all sorts of work within the body. So the mitochondria are membranous organelles. They are surrounded by a double unit membrane. The inner membrane folds are called cristae, and this is where the main functionality of mitochondria originate, because ATP is synthesized by enzymes on these inner membrane folds. And again, the process of this occurring is called cellular respiration. So basically we have the conversion of energy in fuel molecules converted into the energy of ATP, which is what our body freely spends anytime work is done in the body. So pretty much any form of work aside from things that don't require energy, like passive transport, our cells are going to be utilizing ATP to do those. So the space between the cristae are called the matrices, the matrix. This is going to contain ribosomes, and the really cool thing, in my opinion, about mitochondria is they have their own DNA that's separate from the nuclear DNA, and that's just known as mitochondrial DNA, abbreviated mtDNA. Mitochondrial DNA is passed along the maternal lines, so it's a really unique tool that anthropologists and others who study the human body can learn about genetics and basically trace back a lineage through the mitochondrial DNA. Okay, so the rough endoplasmic reticulum, or the RER as it is sometimes abbreviated, endoplasmic reticulum, typically just abbreviated ER. If we want to specify that it is rough endoplasmic reticulum, sometimes it's referred to as RER. What this is, it's another membranous organelle. It's basically parallel flattened membranous sacs that are covered with ribosomes, and it gets its name from the fact that ribosomes are actually attached to it. It gives it a more rough appearance than we see with the smooth endoplasmic reticulum, which I'll talk about momentarily. So it is continuous with the nuclear envelope and with smooth endoplasmic reticulum, so it basically forms this continuous network, which makes sense because what the rough endoplasmic reticulum does is it synthesizes proteins that are packaged, and we of course have this flow of information from DNA to RNA to proteins, and to do that we have to have the nucleus, ribosomes, and the endoplasmic reticulum to actually package those proteins and get them ready for use within the cell or export to other cells within the body. So rough endoplasmic reticulum is particularly concentrated within cells that have to produce a lot of protein, such as our digestive glands. So for example, our salivary glands, we're producing enzymes that will actually help break down food. And enzymes, of course, are proteins. So we're going to see a lot of rough endoplasmic reticulum within those glands. It also makes phospholipids, primary component of cell membranes, and proteins that are found within the plasma membrane. Okay, the smooth endoplasmic reticulum. This again is continuous with the rough ER and the nuclear envelope. Very similar, except it lacks ribosomes. That's why it's called smooth endoplasmic reticulum. The cisternae are more tubular and branching than what we see in the rough ER. And what the smooth ER does is it synthesizes membranes, steroids for the reproductive system, for example. It will synthesize estrogen and testosterone, for example. Also lipids. Lipids are just fats. Waxes and steroids. It also functions in detoxification. So you'll find a lot of smooth ER within the liver and the kidneys. And it also functions in calcium storage. And calcium it's important to keep calcium sequestered within the body, and it's only released when we need it. So it's really important for skeletal and cardiac muscle because calcium is going to play a crucial role in muscle contraction. So this illustration here just shows our smooth endoplasmic reticulum and rough endoplasmic reticulum. Again, the rough is going to actually be studied with these ribosomes, giving it a slightly altered appearance from the smooth endoplasmic reticulum. And another thing I want to mention with the detoxification purpose of smooth ER is basically our smooth endoplasmic reticulum is very efficient at detoxification. What happens when you have somebody develop a tolerance to a certain drug, whether that be an illicit substance or a prescribed medication, if a person is building up tolerance, basically what's happening is our smooth endoplasmic reticulum is becoming more and more efficient at breaking down that substance, that drug. So you eventually need to take more and more of that drug to feel the same effects because our body is just getting better at breaking it down. Here are some images of endoplasmic reticulum taken with the aid of an electron microscope. You can see here, it's kind of difficult to see, but the ribosomes on the rough ER kind of give it this beaded appearance, and then smooth endoplasmic reticulum over here. And this is just an inclusion, which I mentioned towards the beginning of this lecture. Inclusions are not organelles themselves. They're typically just stored products within a cell. In this case, it's an oil droplet. Okay, ribosomes. Ribosomes are one thing that you will find in prokaryotic cells as well. They are responsible for manufacturing proteins. They themselves are made up of protein as well as RNA, ribonucleic acid, which is one of two types of nucleic acids found in the body. They are found in nucleoli, free in the cytosol, just meaning they are distributed throughout the cytosol or fluid of the cell, and then of course on the rough endoplasmic reticulum as well. What ribosomes do is they use directions encoded within messenger RNA or mRNA to assemble amino acids into proteins, and the sequence of amino acids is going to be specified by the order of nitrogenous bases within our genetic code, which is of course DNA. So if you haven't studied the process of transcription and translation in a while, that's okay. You may want to look into chapter 4 of the Saladin text to kind of have a quick refresher on that process. This is a critically important process for human biology, but since A&P is a little bit more body system oriented and specific for those going into allied health and nursing, it's really kind of something that I don't cover in this class. It's kind of focused on subjects that are on a need-to-know basis, and because most of us are not looking to become geneticists or microbiologists, it's something that I don't really cover in this class. But I do want you to be familiar with the fact that, you know, our genetic code is ultimately coming from the nucleus, the DNA. It's going to be dictating what traits we have via the specific sequence of nitrogenous bases. And the way that we make our genetic code into something that's actually functional, into the structures and hence functions of the body, is through building proteins from that genetic code. So it's something that you should be familiar with the basic process of. Okay, so the Golgi complex, what the Golgi complex is it's a system of flattened sacs called cisternae. It synthesizes carbohydrates, packages proteins, and glycoproteins. And a glycoprotein is basically just a combination of a carbohydrate and a protein. So it will have a carbohydrate moiety and a protein moiety. It also forms vesicles. This is important for lysosomes, secretory vesicles. If we're secreting products from a cell, they can be transported via vesicular transport. It also makes new plasma membrane. So the Golgi complex kind of looks similar to endoplasmic reticulum. In most images you see it's a little bit more compact or flattened looking. So this is the Golgi complex, and we'll actually see the products that it produces are going to be transported in what are known as Golgi vesicles. Okay, moving on to lysosomes. Lysosomes are basically packets of enzymes within a single unit membrane, so it's not a double membrane, it's just a single membrane. They are quite variable in shape. The functions of lysosomes, primarily intracellular digestion of large molecules. So they're basically, sometimes the analogy that people come up with is they're like the little stomachs within a cell. So they basically digest worn out organelles, autophagy or autophagy. Anytime you see that auto in front of a word, that means self. Autobiography, for example, autopsy actually. So auto means self. Digestion of worn out organelles, it's not destroying a foreign particle or foreign material in that case. It's actually basically digesting parts of the body itself. But that can be a normal process because, of course, our cells typically will have a lifespan that's less than our whole organism lifespan. They're also important for this process of autolysis. Again, self. Lysis is to break. So autolysis or autolysis, programmed cell death. For example, during and immediately following pregnancy and birth, the uterus increases in mass quite substantially, and then several weeks after having given birth, a woman's uterus will shrink, and that is due to autolysis. So programmed cell death, that can actually be a natural process in the body. And basically what lysosomes do is they break down stored glycogen in the liver to release glucose. That's another one of their functions. So glycogen is basically like starch, except it's for animal cells. It's a way to store potential energy, and then when we need that, we can break that down into glucose with the help of lysosomes. Okay, peroxisomes. These are smallish organelles that tend to resemble lysosomes, but they contain different types of enzymes. They are produced by the endoplasmic reticulum. They're in most cells, but they are more abundant in liver and kidney cells. And they function in neutralizing free radicals. Those are molecules that can cause cellular damage. And they also detoxify alcohol, other drugs, and toxins. They use oxygen to oxidize organic molecules, which that produces H2O2, which is hydrogen peroxide, as a byproduct. And catalase, which is found in the peroxisomes, that's a special type of enzyme that will actually decompose hydrogen peroxide into water and oxygen, rendering it harmless because hydrogen peroxide can actually cause cellular damage as well. They also break down fatty acids into acetyl groups for use by the mitochondria. So a couple of images taken with electron microscopes here. We have our mitochondria. They are actually pretty variable in size and shape, and they're actually quite mobile within the cell as well. So our mitochondria, typically we think mitochondria is kind of kidney bean shaped, but as you can see here, There are variations in the size and shape. Here's our Golgi complex or Golgi apparatus. And then peroxisomes. Peroxisomes here are basically kind of nondescript globular organelles. Here we have some smooth endoplasmic reticulum, and of course we have our lysosomes here. These darker small shapes here are the lysosomes. Okay, so switching gears a little bit, I'm going to be talking on a few select topics from Chapter 4 of the Saladin text. The cell cycle is basically what occurs throughout the cell's life cycle. So cell cycle, think the stages that a cell goes through from the time it's formed to the time that it reproduces or dies. So we have several different phases of the cell cycle. A lot of people think of the cell cycle as being just mitosis, but mitosis is just one small part of the cell cycle. And it's actually not going to be the majority of the life cycle of the cell. The majority is spent in what's known as interphase, which is part of the cell cycle, but not part of mitosis. So we have what's known as the G1 phase, first gap phase. This is when the cell is accumulating materials that it needs to actually replicate its DNA. Replication of DNA is just creating an exact copy of DNA based on the principles of complementary base pairing of those nitrogenous bases, adenine, thymine, guanine, and cytosine, or A, T, C, and G. Then we have S phase, which is referred to as S phase because it's the synthesis phase. This is where we actually have the DNA replication occurring. So again, we're basically just creating an exact replica of the DNA molecule, barring mutations that might occur, of course. And then G2 phase is the second gap phase. We have replication of centrioles, and those are going to be really important for cellular movement, microtubule movement. They're basically going to kind of orchestrate the process of mitosis. And we also have enzymes being synthesized that are important for actual cell division. And then the M phase or mitotic phase, if a cell successfully goes through gap1 phase, S phase, gap2 phase, then it may enter the mitotic phase or M phase. And again, a lot of people mistake mitosis as being synonymous with the cell cycle, but in fact, mitosis is just one small part of the cell cycle. Mitosis itself is basically division of the nuclear material, the DNA. And then cytokinesis is when you actually have the cell splitting into two daughter cells, and that's cytoplasmic division. So division of everything aside from the actual DNA. And then the gap 0 phase is what happens when cells have actually left the cycle. And this might occur when we have, for example, a mature spinal cord or brain cell. So we do have some types of cells that once they are mature, they don't actually divide again. And the cell cycle duration will vary between cell types. So, we have some cells that proliferate quite quickly, such as the cells that line our stomach. They're exposed to our stomach acid. They have a pretty fast turnover rate. Another example of cells that have a fairly fast turnover rate would be our skin cells. And then, like I said, there are some cells that, once they're actually mature, they will not divide. So a mature neuron, for example, that's why we can have injuries resulting in paralysis, because those cells can't just replace themselves via mitosis. So this is basically, it's kind of like a little pie chart here that shows a typical cell cycle: G1 phase, synthesis phase, G2 phase, and then mitotic phase. Okay, Okay, so mitosis and cytokinesis, this is the part of the cell cycle that kind of gets most of the attention in most of your typical biology classes. It's the process wherein one cell's duplicated genetic material divides and the cell splits into two daughter cells. So technically speaking, the genetic material itself dividing is mitosis, and the cell splitting into two daughter cells is cytokinesis. The functions, there's many different functions that are imperative for human biology. One is just embryonic development. So we all develop from a fertilized egg. That's basically one cell with a complete set of DNA. And in order to grow into a multicellular organism, that one cell will divide into two cells. Those two cells divide into 4 cells, etc., until you get the trillions of cells that make up a mature human. Tissue growth, so for example, throughout childhood, we are adding mass to our body through the process of mitosis. Also, replacement of dead cells. So like I mentioned earlier, the typical cell life cycle will be, or the cell lifespan will be less than our organism lifespan. So, we might live to be, if we're really lucky, we might live to be, 90 years old. Most of our cells do not live that long. And then repair of injured tissues. And most of our tissues can repair themselves to some extent. Some injured tissues aren't able to really get back full functionality, and we might just have replacement of what was functional tissue with scar tissue, for example. But many types of tissues can effectively repair themselves following death of cells. So I would like to cover the phases of mitosis, which is just the nuclear division. The phases of mitosis are prophase, metaphase, anaphase, and telophase. And then typically cytokinesis occurs following telophase. So PMAT is an acronym that I like to tell my students. It's fairly easy to remember. Prophase, metaphase, anaphase, telophase. And that just is the order of phases of mitosis. Okay, so timing of cell division, when do cells actually divide? Well, cells will divide when, now they don't always divide when these conditions are met, they just can divide if these conditions are met. So they can divide when they have enough cytoplasm to partition into two daughter cells during that process of cytokinesis, when the DNA has been successfully replicated during S phase of the cell cycle, when they have an adequate supply of nutrients, when they are stimulated by growth factor, when there's open space due to neighboring cell death, and cells will stop dividing when we have a loss of growth factors or nutrients, when we have what's known as contact inhibition, which all that is if you have cells right next to one another, they're in contact with one another, that's contact inhibition. If there's not an empty space, there's basically no space for this one cell to actually divide and form two daughter cells. So that would be contact inhibition. There's no need for that division. Okay, let's look at the process of mitosis itself. This is going to be, of course, a very brief overview of mitosis. I haven't really talked about chromosomes. A chromosome is basically the condensed version of DNA, and a typical human cell, of course, is going to have 46 chromosomes, 23 from each parent. So 23 from an egg cell, 23 from a sperm cell, that's going to equal 46 total chromosomes. Now, for the process of mitosis, we will have had the S phase occur prior to mitosis during interphase. So we come up with actually 92 chromosomes. That's then going to be split in half during the process of mitosis and cytokinesis, such that each daughter cell will end up with 46 chromosomes once more. So we have DNA replication, that leads to 92 chromosomes, and then division, which again leads to 46 chromosomes. So prophase, just remember this acronym PMAT for the series of events that occur here: prophase, metaphase, anaphase, telophase. With prophase, we have the chromosomes condensing. The nuclear envelope, which is of course that partition around the DNA, is actually going to break down because we have to have a way to actually partition these duplicated chromosomes on opposite ends of the cell in preparation for cytokinesis. We also have spindle fibers that grow from the centrioles, and the centrioles will migrate to opposite poles of the cell. Metaphase is one of the most, I guess I would call it kind of an obvious phase. It's typically pretty easy to pick out if you're looking at an onion root tip slide, for example, because all of the chromosomes are lined up in the middle of the cell. So we have chromosomes that lie along the midline. Some of the spindle fibers attach to what are known as kinetochores. And then the fibers of the asters will attach to the plasma membrane as well. So the spindle fibers are basically just these cord-like proteins here. So they are going to be attaching to the plasma membrane and also attaching to the centers of these duplicated chromosomes that have lined up in the middle of the cell. Anaphase, this is where we have initial separation of the duplicated chromosomes. Duplicated chromosomes are referred to as sister chromatids, and then once they're actually separated from their identical pair, they're again referred to as chromosomes. So anaphase, you have the centromeres dividing in two. The spindle fibers are going to pull those chromosomes apart to opposite poles of the cell. And then each pole, which are the future daughter cells, will have an identical set of genes, because all we've done is we've separated the identical chromosomes from one another. So we're setting up to basically just have two genetically identical daughter cells. And then telophase, the chromosomes gather at each pole of the cell. The chromatin will decondense. The new nuclear envelope will form, so we have to basically repartition the DNA from the rest of the cell content, so we have the formation of new nuclear envelope. The new nucleoli appear in each nucleus, and the mitotic spindle will vanish. And then cytokinesis, of course, basically with an animal cell, we have what's known as a cleavage furrow that will form between these two daughter cells, and the two daughter cells will pinch off from one another until we have two completely separate daughter cells. And of course, by the end of this process, each of those daughter cells is going to have its identical set of DNA. So that is mitosis in a nutshell. I know this was a very brief introduction to mitosis, but like I said, hopefully it is somewhat of a review for most of you. Of course, as always, let me know if you have any questions about any of the topics that I've covered in this lecture, and thank you for listening.